Namaskar, dear learners. Today we will discuss about the electron microscopy. This topic is from the cell biology BBCCT 103 of the B.Sc Honors Biochemistry program, and this is also the topic of the course Bioanalytical Techniques MBC-003 of the MSc Biochemistry program. And this is an important topic in the field of the microscopy, cell biology, toxicology and medicine. And electron microscopy is an important tool which is widely used in the lab, especially in the research labs. So today we will discuss about the electron microscopy, what is electron microscopy, what are types, and what is the working principle of the electron microscopy. So in this session I will cover about the background, history, working principle and types of electron microscope, and we will discuss about the transmission electron microscope and scanning electron microscope, and explain what is the mechanism behind to produce the highly resolved image by the electron microscope, and what are the steps of the tissue fixation of the biological specimen which is required for the electron microscopy, and what are the comparison between optical microscopy and electron microscopy, and what is the application of the electron microscopy. So why we need a electron microscopy? Firstly, the microscope is of two types based on the source of the illumination of the image. So basically the using of the source of illumination, light microscopy and the electron microscopy are the two types of the microscope. So the microscope that use the electron, which is called the electron microscope, the microscope use the light source as a illumination of the sample, which is called the light microscopy. In previous session we have discussed about the light microscopy, which is also called the optical microscopy. So in optical microscopy we use the condenser lens, objective lens, and the image is formed by the interaction of the light with the specimen. So here in electron microscopy it's quite different from the light microscopy. So today we will discuss about the electron microscopy. If you recall the previous session, which is based on the optical microscopy, there are different kind of the optical microscopy are using in the lab, especially in the biological lab, like bright field microscopy, dark field microscopy, phase contrast microscopy, phase fluorescence microscopy. So number of the different kind of the microscopy, light microscopy are used to view of the different kind of the biological specimen, not only biological specimen, like the cells, bacteria and cell structures, because this kind of the cells are tiny and small in size, so human eye cannot see without the microscope. That's why we need a microscope. In our surrounding there are so many kind of the tiny microorganism available within the body and outside of the body, like the in water, in the earth. There are so many kind of the microorganisms available. To study of that microorganism and the structure of the cells, we require the microscope. So in the level of the size, in the level of the morphology of the cellular structure, we require the scientific equipment such as the microscopy. So you can see here the overview of microscopy. So in light microscopy that has the limitation, because in light microscopy light use the light source. So if we see the resolving power and the magnification in the light microscopy, there is a limitation. So the limitation of the resolving power and the magnification led to the deployment of the electron microscopy, because electron microscopy gives a very deeply view of the internal structure of the cells, viruses and the bacteria. So it surpasses the resolution limit of the light microscopy. So this is the overview of the microscopy. So here we will discuss about only electron microscopy. As in previous session we have already talked about what are the types, principle of the light microscopy. So what is an electron microscope? As we discussed here, the electron microscope uses the light instead of the uses of the electron instead of the light to view of the biological specimen. and it offers the high resolution magnification of object and nanometer scale. It means the wavelength of light is very small, and its wavelength is 0.002 nanometer. So it is higher than the wavelength of the light. So we can see the wavelength of electron is very smaller than the light wavelength, and it allows scientists to observe structure as well as the individual atoms, making it indispensable in fields such as biology, material science, and nanotechnology. So the electron microscopy has wide scope in every field of the scientific study, like engineering, medicine, toxicology, and clinical biochemistry. So the wide spectrum of the application electron microscopy used in the research field. And if we want to study of the cellular structure like subcellular organelles like mitochondria, endoplasmic reticulum, nucleus, so we need a electron microscope to study of that kind of the structure at the molecular level. So early theories and limitation of optical microscope led to a development of the electron microscope. So optical microscope has the limitation, has the limited resolving power with magnification. If we see the magnification, the light microscope can up to magnify the smaller object up to the 2000x, and the resolution is 200 nanometer. But in case of the electron microscope, using the electron, that can magnify the smaller objects, smaller object up to the 0.002 nanometer, or we can say the 1 angstrom nanometer size. So the wavelength is so smaller as compared to light wavelength. So we can see visual light is used in light microscopy. The range of the light wavelength is 400 to 700. So we can say that in electron microscopy the wavelength of electron is 0.004 nanometer. So we can scan the smaller object that can up to the 0.004 nanometer in the electron microscopy. So the development of the electron microscopy because of the limitation of the optical microscopy. If you are a researcher or the biochemist, you want to study of the cell structure, internal structure of the cells, internal structure of bacteria, internal structure or external structure of the viruses, we use the electron microscope. So here you can see the electron microscopic view of the cellular organelles and cells. So these structures are captured by the electron microscope. So if we take a thin slices of the biological specimen, processed specimen slices by the fixative, emitting, sectioning, and if we want to observe these kind of the biological specimen under the electron microscope, we can see this kind of the structure of the Golgi body and the nucleus. So here you can see the Golgi body is a vesicle type of the Golgi body structure inside the cells. So this kind of the structure within the cell we cannot see using the light microscope. We can only see this tiny structure using the electron microscope. So here you can see the nucleus. Here this is the nucleus, then this is the nuclear membrane and endoplasmic reticulum and the mitochondria. So these structures are provided by the electron microscopy. So here another figure is the electron microscope view of the endoplasmic reticulum and mitochondria. So this is this kind of the subcellular organelles are present in the cells, are present in the inside the cells. So these specific kind of the organelles present in the eukaryotic cells. So these kind of the subcellular organelles have the specific kind of the functions, and you can see the different kind of the morphology we can also observe under the electron microscope. So I know you have gone through the structure and function of this kind of subcellular organelles in the cell biology course while studying this course in that BBCT 103. So the electron microscope is invented by the two famous scientists Max Knoll and Ernst Röska in 1931. So the first time the discovery of electron led to the development of the electron microscopy, because the J.J. Thomson discovered the electrons, and another the scientist de Broglie also proposed the electrons have a wave-like theory. So using this kind of the discovery led to the development of the electron microscope by the Max Knoll and Ernst Röska. These two scientists together worked and built the early type of the transmission electron microscope in 1930s. So Ernst Ruska demonstrated that electron's beam can be used to magnify object at a much higher resolution than light microscope. So, and he got the Nobel Prize due to his extraordinary work to invent this kind of the electron microscope. So this is the first scientist which developed the electron microscope. This is the physical sciences. So इलेक्ट्रॉन माइक्रोस्कोप दिखता कैसा है, इसके लिए हम देख सकते हैं दिस इज द व्यू ऑफ द एक्सटर्नल व्यू ऑफ द इलेक्ट्रॉन माइक्रोस्कोप. सो इलेक्ट्रॉन माइक्रोस्कोप इज लाइक अ cylindrical column, control panel and computer. There are three things we can see in the lab. So cylindrical column, inside the cylindrical column there are specific kind of the lenses are used in that, and the control panel is the controlling of the beam of the electrons source, and the whole instrument is connected to the computer screen, and computer screen provide the image of the biological specimen using the electron. Now we discuss about what are the main parts of the electron microscope. So in electron microscope, the source of electron is the electron gun. So there are, here you can see the electron gun is focused beam of electron is directed onto the sample. So here electron gun is made up of the tungsten filament cathode. So tungsten filament cathode is electrically heated voltage at 100 to 1000 volts, and it produces the electrons. So the electrons are accelerated with the help of the anode, and so in that column, cylindrical column, electrons are produced like the beam of electrons in, and these electromagnetic lens are parallel arranged to focus and form the maintain of the electron beam onto the sample. So that electron gun, electromagnetic lens, and then specimen holder. Specimen holder is a kind of the holder that we keep the sample on that area in the electron microscopy. Another important part is the electron detector. So electron detector detects the electrons coming from the biological specimen. So it helps to detect scattered electrons coming from the specimen. And then another part is the image viewing and recording system. So the final image is projected on a fluorescent screen or the computer screen, and the whole process of the electrons movement in the chamber are the vacuum condition. So vacuum condition is required to move up the electron in the long cylindrical column. So you can see here, and this is the image of the electron microscope. Here the vacuum is created by the using of the high vacuum pump. So high vacuum pump is required, is attached to the electron microscope to create a vacuum. So vacuum provide the condition to movement of the electrons down to the electrons onto the biological sample, and the other components like electromagnetic lenses, electron detectors are provided here to maintain the electron beam in a direction that it falls on the biological specimen. So what is the working principle of electron microscope? Basically, the principle of electron microscopy underlies on the interaction of the electrons with the biological sample. So, so the light can be absorbed, reflected, and scattered. So on the basis of the electron with interaction of the biological specimen, electrons say some kind of information of the biological specimen, and then it detected by the recorder or detector, and then finally produce a magnified and highly resolved image of the biological specimen. So here you can see the interaction of high energy electrons with matter in electron microscopy. And here होता क्या है कि जब electrons interact with biological space, there are four kind of things can be happen. Here, when the incident beam, the incident light electron means the light that hits the biological specimen is called the incident beam, incident electron beam. So that within the biological specimen or object have the thick and thin density of the material. So when the electron interact with the different thickness and thinness of the medium of the biological specimen, some kind of the electron are not scattered, but some kind of the electron can go into the nuclear level of the atom. The electron are tiny, having the smaller wavelength, so it can interact with nuclei without losing of the energy. But some kind of the electrons, when they interact with the different kind of the density, or we can say the refractive index of the biological specimen, and their direction and the velocity are changed. So this kind of the scattered electrons are very important to formation of the image in the electron microscope. So, and some kind of electrons are only interact with the outer surface of the electrical orbital of the atoms. So this kind of the electrons initiate the electrons, and some kind of electrons are moving from their electrical orbital and they goes to the out of the electrical orbital of the atoms. So this kind of the electrons are called the secondary electrons. यहाँ हम इस तरीके से समझ सकते हैं, जब कोई कुछ इलेक्ट्रॉन की बौछार किसी बायोलॉजिकल स्पेसिमेन पर गिरती है तो उसमें से कुछ इलेक्ट्रॉन्स होते हैं जो इंटैक्ट होते हैं स्पेसिमेन बायोलॉजिकल स्पेसिमेन के मटेरियल से, बट वो डिफ्लेक्ट नहीं होते हैं, ना तो कोई एनर्जी गेन करते हैं ना वो एब्जॉर्ब होते हैं. लेकिन कुछ इलेक्ट्रॉन ऐसे होते हैं जो एटम के न्यूक्लियस लेवल तक जाते हैं, बट उनकी एनर्जी कुछ चेंज नहीं होती, बट उनकी डायरेक्शन होती है वो चेंज हो जाती है. बट कुछ इलेक्ट्रॉन ऐसे होते हैं जो एटम के लेवल पर जाकर के इलेक्ट्रॉन से इंटरेक्ट करके कुछ इलेक्ट्रॉन को बाहर निकाल देते हैं. सो दिस काइंड ऑफ इलेक्ट्रॉन्स आर सेकेंडरी इलेक्ट्रॉन विच आर यूज्ड फॉर द स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप. बट इन केस ऑफ द ट्रांसमिशन इलेक्ट्रॉन माइक्रोस्कोप, दोज इलेक्ट्रॉन्स आर स्कैटर्ड from the specimen. They are captured in the electron microscope and produce the image of the biological specimen. So in that case, the scattered electrons are very important in transmission electron. If the high refractive index of the biological specimen, they produce the gray or the dark image of that particular area. अगर इस बायोलॉजिकल स्पेसिमेन का जो स्ट्रक्चर है उसके अंदर ट्रांसपेरेंट स्ट्रक्चर है तो it produce the brightness of that structure. So here what are the type of the electron microscopes? So based on the biological interaction of the electron with the specimen, it produce the highly resolved image. So here now I come to the discussion of the types of electron microscope. So transmission electron microscope and scanning electron microscope are two types of electron microscope based on their functions or the role of the electron microscope. So transmission electron microscope, it passes electrons through a specimen. Basically a transmission electron microscope is based on the scattering of lights from the specimen and produce the highly resolved image. So in that case the transmission electron microscope provide the 1 lakh to 2 lakhs higher resolving image or the magnification. So it is basically used for the study of the internal structure of the subcell organelles, bacteria and the viruses. And the scanning electron microscope are based on the emission of the secondary electrons from the biological specimen. So the scanning electron microscope scan the outer surface of the biological specimen. So here the two types of electron microscope based on their functions. Now I come to the discussion of the what are the basic part of the transmission electron microscope. As we see here, the electron microscope is a cylindrical tube, the long tube. Inside the cylindrical tube, that the different kind of the magnetic lenses, condenser magnetic lenses, electromagnetic objective lens and electromagnetic projector lens here inbuilt in this system. So here there is a parallel kind of the lenses are arranged in position of the biological specimen. So here the electron gun are generated the electrons, and another is the anode which accelerate the electrons speed in the form of the electron beam. So the electromagnetic condenser lens focus the electron beam onto the specimen, the prepared specimen. So Here the specimen are so thin, like 50 to 100 nanometer in size. We prepare the biological specimen is a different way. This is the complex process of the biological specimen in the electron microscopy as compared to the preparation of the biological specimen in the light microscopy. So when the electron gun produce the electrons and with the high voltage, and electrons are accelerated with the help of the anode. Anode is the positive charge. The electrons are negatively charged. So beam of electron is passes in a straight direction, and it goes through the electromagnetic lens. So electromagnetic condenser lens focuses the electron beam, and it passes the electron beam as a gun in the high speed onto the biological specimen. Here the electron beam are interacted with the biological specimen, as we discussed in previous slide, that the interaction, the electrons are interact with the biological specimen, and the scattered electrons coming from the biological specimen further captured by the electromagnetic objective lens. So electromagnetic objective lens further magnify, and these kind of electrons are passes through the projective lens, and then finally electron beams having the information of the biological structures, and then finally fall on the fluorescent screen. Here in electron microscopy, the electrons are finally reached to the fluorescent screen, and the fluorescent screen have a specific kind of the detector, the positive grid like detector, and it is connecting to the computer system. So here the image recording system captures the electrons information and passes to the computer system, and in the computer system the image of the biological structure is visualized here. So basically in transmission electron microscope produce the internal structure of the biological specimen. If the biological specimen in the eukaryotic cells like hepatocytes, then it produce the internal structure of the eukaryotic cells like subcellular organelles, nucleus, mitochondria, endoplasmic reticulum. What are the structure? If the biological specimen have that some kind of the diseases, the toxicity in the cells, then the structure is different from the normal cellular structures. So here you can see the two kind of the figures here, that one is captured by the light microscope and another is the electron microscope. So this is the specimen of the hepatocyte cells. The hepatocyte cells is present in the human liver. So this is the structure of the hepatocytes you can see here, and this is the 400x magnification, and this another image of the electron microscope of the hepatocyte here. So you can see the difference of the both images. One is captured by the light microscope, another is the electron microscopic image. So here you can see the different kind of the subcellular organelles in the cells like nucleus, mitochondria, cytoplasm, nuclear membrane, and the endoplasmic reticulum. Because of it produces a high resolved image at the atomic level. So we can see that before the invention of the electron microscopy, we cannot see this kind of the structure, and only the light microscope is able to provide the biological object at a 200 nanometer sizes. So here you can see this is the image of the light microscope. You can see the hepatocytes, nuclei, and what kind of the hepatocyte are arranged in the tissue level. So these are two different kind of the image which can differentiate based on the observation of the light microscopy and the electron microscope. So here, and you can also compare the different components of the transmission electron microscope and the light microscope here. So in light microscope we use the lenses made up of the glass, condenser lens, objective lens, and eyepieces. But in case of the transmission electron microscope, lenses are made up of the magnetic coil, or we can say the electromagnetic coils here. And the source of the light, source of the illumination of the sample electron in electron microscopy, light in the light microscopy, and the sample preparation is different in both microscopy, electron microscopy and the light microscopy here. And we can also study of the living, non-living cells in the light microscopy, but in electron microscopy we only see the object in a dead state level, because electrons kills the living organism if we observe under the electron microscopy. So now another microscope is the advanced level of the electron microscope, is the scanning electron microscope. So here electron microscope, especially scanning electron microscope, is used the scanning of the surface of biological specimen. So this kind of the image is produced by the detection of the secondary electrons coming from the biological specimen. So in that case, the low voltage is applied to produce the electron beam, and this kind of electron beam, under the passing of the magnetic condenser lens, electromagnetic objective lens, and then this kind of electron beam is only scan the surface of the biological specimen. So this scanning electron microscope is ideal for study of the outer layer of the biological specimen, outer layer of the cells. So you can see here the made up of the construction of the scanning microscope is the transmission scan microscope, and the scanning electron microscope is similar, but the basis of the produce of the surface image is based on the secondary electrons here. So this scanning my electron microscope is based on the secondary electrons which interact with the biological specimen. And this is the image of the blood cells which show like the Y convex shape. So this kind of the blood cells surface, we know that is the biconvex shape is produced by the scanning electron microscope. And different kind of the matter, the object, we can analyze the surface of the biological cells or membranes here. So before observing of the biological specimen under the electron microscopy, either transmission electron microscope or the scanning electron microscope, it requires the highly complex process of the tissue preparation. So in the tissue preparation, first we fix the cells in the chemical fixative or the freezing, like cryo freezing method. So here if the tissue is the animal or either the plants, we use the osmium tetroxide and formaldehyde. So here the processing like the chemical fixation, tissue processing, sectioning and staining. These are four kind of the tissue preparation required for electron microscopy here. And finally after preparation of the biological specimen, the sectioning required to cut the tissue at the level of the 200 nanometer thickness. So and after that last stage is the staining, and we stain the tissue with the helping of the some different kind of the stains here. So after preparation of tissue, we can view of the prepared the grid of the biological species under the electron microscope here. So that there's some kind of the limitation of electron microscopy. The scientist observe like living specimen cannot observe in an electron microscope because of the high temperature in the column of the electron microscope. Sample preparation is complex process like fixation and the coating and the embedding, and non-living samples must be not observed due to vacuum and electron beam. So because of the damage of the sample by the electron, and the electron microscope is highly expensive. That's why this kind of the limitation here. So we know that electron microscope is a very crucial equipment in microbiology, medicine and clinical biochemistry, and fixation, tissue processing, sectioning and staining are required in the electron microscopy observation. And TEM is used for the internal structure study, and SEM is used for the surface study of the biological specimen. So both are parts of the TEM and SEM techniques are very important in the biological science. So these are the some suggesting readings. For more details about the electron microscopy, and the topic is included in the cell biology course, and you can download in this cell biology course of the B.Sc Biochemistry program. Thank you.